So I'd really like to thank Munir for the invitation and the organizing committee. What I'd like to do is talk a little bit about something that Rory introduced, polygenic risk scores, then give some examples about how polygenic risk scores may actually have some relevance to pharmacogenetics. And then I'll end by talking about a new project very much inspired by UK Biobank called Our Future Health. So in the 20th century, this is how we did genetics, family-based studies, and trying to establish what the single gene causation was in families that suffered from a high burden of a particular condition. But in the last 20 years, we've established that a large component of genetic predisposition to a wide variety of diseases and traits and phenotypes is due to polygenic risk, the accumulation of dozens or hundreds or even thousands of very, very small risks in dozens or hundreds or thousands of genes or intergenic regions that control genes. And the question is, how do we integrate this into clinical practice, and is there a role in pharmacogenetics? So the way all of this came about was the appreciation that when the technologies became available to use gene chip devices to look at, first of all, hundreds of thousands and then millions of variants spread across the genome, it was possible to migrate from family-based studies or candidate gene studies to whole genome studies, where essentially we took epidemiologic studies, case series, case control studies in the main part, and asked the question, are there variants that are either more or less common in cases versus controls? And because we were looking for very, very small effects, we needed massive sample sizes. So one of the things that's still limiting in this new world of polygenic risk is that while we have adequate large sample sizes for common diseases, for rare diseases, we don't have that yet. So we don't have robust polygenic risk scores for some of the rarer conditions. And for the super rare stuff, that is only a few families in the world perhaps, or a few hundred people in the world, we'll never have a polygenic risk score. But for the common causes of the common diseases, we have these, and these have been done by stitching together case control consortia across the world, initially with tens of thousands of cases, now in some cases hundreds of thousands of cases, in order to be able to establish what the variants are that are more or less common in a series of cases than in controls. For quantitative traits, things like blood pressure, all of the wonderful variables that Rory said were in the biomarker panel for UKB, much of the work actually comes from UKB because the only place that you can find a large number of genotype people with some of these biomarkers is UKB, and it wouldn't make sense to try and put that together across other databases. But for most of the major diseases, it's done with international consortia, development of polygenic risk score, and then we take it to UKB and other prospective studies to examine the cumulative incidence of the outcome in a prospective study in people who have high, medium, and low polygenic risk. There is a shortcut to be had. So basically, it took, Doug Easton will be talking about this later in the day, dozens of studies of breast cancer carefully stitched together by Doug and the Breast Cancer Association Consortium to establish the first low penetrance, lower relative risk variants that were associated with breast cancer. Fast forward 10 years, and for the price of 30 seconds on the UKB website, you could actually pull out the same variants because there were prevalent cases, incident cases of breast cancer and the genome array done on 500,000 people, so all of those cases and a much, much larger number of controls. But the major use of UKB is not for discovery, it's for the calibration and establishing what the relative risk is between high and low polygenic risk that we've derived from these large international consortia. So you can find all of these data at the EMBL website. Literally hundreds of conditions now have been subject to genome-wide association studies, and the genome is just painted with variants, and each of these individual colors indicates a different trait or condition. For common diseases like breast cancer, over 100,000 of these low penetrance, lower relative risk variants now robustly established. And we can put these together very simply into a polygenic risk score just by taking all of that data, establishing what the relative risk is for each of the variants, and then for any individual in the UKB or anybody who's had their 23andMe or another genome array run, we can establish whether each individual carries a higher number of the risk variants or a lower number of the risk variants or an average number of the risk variants, and put people essentially on a normal distribution of risk. So does this have any relevance to pharmacogenetics? So those of us who worked in genetic epidemiology, again back in the 20th century, were always supremely envious of you guys in pharmacogenetics, because you pretty much knew for your drugs what the targets were, how they moved around the body, pharmacokinetics, and how they got metabolized either to active metabolites or to be excreted. And so you could, back in the dawn of genetic epidemiology, carefully screen the genes that were relevant here, look at the between-person variation, and ask, does that between-person variation translate into different pharmacodynamics, different pharmacokinetics, and ultimately you were very successful in establishing for a wide variety of drugs variants that collectively were associated with differences in efficacy or differences in toxicity. We didn't have, for most diseases, these metabolic pathways to examine, and one of the revelations from common disease genetics using genome-wide approaches is that many of the variants we find are not in genes, or they're not in a P450, and they're not in a metabolizing gene for a biomarker that we thought might be relevant to the disease. But they're in what used to be called gene deserts, and presumably they're involved in controlling the activity of other genes. And we never would have been able to find these using the candidate gene approach that was so successful for pharmacoepidemiology. So a couple of examples immediately came along where, in fact, a genome-wide approach did identify things of potential relevance in pharmacogenetics. This is one of Rory's studies, doing a genome-wide association study in one of the statin trials. It became apparent that a variant in this gene was associated with about a 45-fold increased risk of developing myopathy on a statin. By doing a genome-wide study and looking across all of the variants on the chip, and then just looking at the variant that had the smallest p-value, the variant fell in a gene that is involved in liver metabolism and was very plausibly involved in statin metabolism, but had not previously been the subject of investigation using the conventional pharmacoepi approach. So genome-wide association studies have found a handful of new variants that you might like to know if you're about to put someone on a statin. you might like to know if somebody's having an adverse reaction. What we really haven't sort of looked at much is whether the polygenic risk scores derived from the genome-wide association studies for traits, whether these have any role in predicting drug response or might have a role in determining the initial starting dose or identifying people who might need higher doses or polypharmacy. So let me show you a couple of examples from the UK Biobank. Looking at the question of, first of all, whether polygenic risk scores do predict any aspect of response to common drugs, and secondly, showing an example of how the current approach we take to deciding who should have a discussion about being on a statin might be altered by knowledge of polygenic risk. So we looked at this in the UK Biobank. We just took 55,000 people who were on one or more antihypertensive medications. So these were people who had been established to have hypertension and their GP had put them on an antihypertensive. And the endpoint here is systolic blood pressure. We also looked at diastolic blood pressure, had very similar findings. Systolic blood pressure at baseline, so on enrollment, the UK Biobank population was put through about 3 or 4 hours of testing, and a very, very reliable, well-calibrated blood pressure was taken. Similarly, as Rory said, the biomarkers included LDL cholesterol, and so looking at 33,000 people on statins, we were able to ask the question, does PRS have any relationship to levels of LDL cholesterol on treatment? And here's the data for blood pressure. So basically, from quintile 1 up to quintile 5 of the polygenic risk score for systolic blood pressure. So these are people who would be predicted to have a lower blood pressure. These are people predicted to have a higher blood pressure. Everybody's on at least one blood pressure medication. And there's a relative risk of about 1.7 for still being hypertensive, over 140 over 90. This is systolic, so over 140, but we see the same result for diastolic. So people who come into essentially blood pressure treatment who are at higher polygenic risk of having hypertension are more likely, 1.7 relative risk, to still be hypertensive after they've been put on treatment. And that translates into about a 5 millimeters of mercury difference in systolic blood pressure, which is about what you get if you put someone on 12.5, a small dose, 12.5 milligrams of hydrochlorothiazide. In terms of the consequences of the blood pressure, so the question is, does the polygenic risk for hypertension among people who are being treated translate into a high risk of cardiovascular events? And the answer is yes, particularly stroke, which is the thing that you'd expect would be associated with blood pressure. So about a 20% higher risk of stroke over the course of the UK Biobank follow-up period if you're in the highest quintile of systolic blood pressure, PRS, even though your doctors put you on treatment. And so really this is, you could say this is a sort of little bit of indictment about the way we practice medicine. We often put people on drugs, but we don't necessarily carefully calibrate the response, or we just assume that the response will be adequate. One GP described this to me as the phenomenon of fire and forget. So I put you on an antihypertensive. If you're otherwise well, I'm not seeing you very often. And by the way, we've had COVID for 2 years, so I haven't seen you at all. And so there's a lot of people who are still hypertensive on antihypertensives. The PRS plays a role in predicting those people. And a very similar story with statins. So basically, almost a threefold increase in LDL cholesterol in people in UK Biobank who are on a statin. So they'd been identified as needing a statin but the LDL cholesterol had not been titrated against statin type or dose, and that translates into about a 15% increase in LDL cholesterol among people on a statin. So that's about the same impact you get with one of the older original statins like simvastatin. Here we didn't see a convincing increase in risk of the cardiovascular endpoints, perhaps because it takes longer to translate into these cardiovascular endpoints, whereas the relationship between blood pressure and stroke is a little more immediate. So these are the findings. The question is why? Well, one possibility is that there's something in the PRS, there are components in the genome that predict in a sort of classic pharmacogenetic way people who won't respond to the drugs. I think it's much more likely that it's just the people who had a higher genetic predisposition to systolic blood pressure or LDL cholesterol started off higher. You give them the same dose, you don't sort of titrate much, and so they wind up at a higher level than the people who had the lower starting values. But this is a cross-sectional study, so we can't establish that. But it does seem that knowing the PRS, if you had it in your database, might identify people who need closer follow-up and are more likely to need higher doses or combination therapy. Another common question is, well, how do you get on a statin? Well, you get on a statin because someone runs an algorithm like QRisk3 and calculates your heart disease risk over the next 10 years, and you qualify according to the expert guidelines for the statin conversation. And if you fall beneath that 10-year risk, maybe we don't have that conversation. We can all recognize that that's a somewhat sort of arbitrary procedure, but that's the way we practice medicine. This is data done with Peter Donnelly's group at Genomics PLC, again using UKB, and taking the established clinical, in quotes, risk predictor, in this case, not QRisk, but the American Heart Association pooled equations. So basically it takes age and sex and BMI and smoking and cholesterol and calculates a risk, family history. And the question is, if we added the PRS information, which is largely independent of those classic risk factors, do we identify more than a trivial number of people who we might give different advice to? And we summarize that with the net reclassification index, which you can think of as the sum of all of the people who become cases, again in UK Biobank, so for whom we correctly predicted high risk, minus the people who, when we added the PRS, we might have moved them below the risk threshold, and then we add that to the controls, who we reclassified when we added the PRS. They were controls, they didn't develop the disease, so did we move more of those controls into a high risk category or a lower risk category? All of this was evaluated for young, old men and women. I won't show you all of the data. So here are the data for the top 5% of risk of coronary artery disease incident. The bottom 5%, blue, is the average in terms of years since assessment. So up 10 years of follow-up now in UKB. First thing to note, obviously men, much higher risk than women at younger and older age with respect to the UKB starting population. Again, this is older, so this is on a different scale. This is up to about 15% of risk. This is only up to about 8% of risk. So at young age, even if you're male in the top 5% of PRS, it's actually quite hard to get above the 7.5% over 10 years risk that is the cutoff. But you will get above it if you are older in that age group, if you're a smoker, obese, have classic risk factors. So this is what happens if you just do the sort of theoretical exercise, just take the AHA only, results very similar for QRisk3, or you add the PRS. This is the risk threshold. Above this risk threshold of absolute risk over 10 years, we're going to have the statin discussion. Below this, we're not obliged to have the statin discussion. Maybe we won't have the statin discussion. So just using the classic risk predictor, about 7.8% of people would be advised to consider a statin. We add the PRS, and most of them would still be advised to use a statin or consider a statin. But about 15% of them fell below the risk because they had a low polygenic contribution to the combined score. Of the 93% of people who would not be advised to take a statin at this younger age, quite a lot of them move up, and because they're at higher polygenic risk, they qualify now for being above that magic 7.5% over 10 years risk. Most people stay in the same category, but some people move up. So you actually, in both men and women, for both young and old, you would make different recommendations by adding the PRS to the classic American Heart Association pooled equation model. And just as proof of principle, let's look at the people who were high previously on the classic risk factor, and it's still high when you add the PRS, that's in the red, low in the green, and the people who wouldn't have qualified but got upregulated over the risk, advised to have a statin, in purple, and people who were downregulated. So there is a difference here, basically higher risk cumulative for people who would only have qualified by adding the polygenic risk information than for people who were down-classified. Obviously, a question, if somebody previously qualified but they don't qualify, do you now not recommend a statin, or do you just keep on sort of adding to the proportion of people to whom you're recommending a statin? That's a discussion for another day, because they are still at elevated risk versus people who are low using both measures. Just a quick note, a lot of concern that most of the data on polygenic risk is generated from European ancestry populations, but it's not true to say that the data don't necessarily predict for non-European ancestry populations. So here's the data from UKB in UK Biobank, smaller numbers for non-European ancestry, but still a very, very similar picture. So I've said all of this. The polygenic risk scores can be derived from a gene chip, fully loaded cost, including bioinformatics, maybe 20 pounds now. And what's going to happen once we've all got this information? You do it once in your life, it will predict risk of all the common diseases and traits. How are we going to cope with this information in medical practice? And the big question is, for all of pharmacogenetics, how do we get this information to the prescriber at the point of prescription? So a lot of what we're talking about with risk communication is risk communication about specific diseases and traits. But of course, it's the prescriber who needs to know and have on their desk at the moment of prescription the information that's germane to pharmacogenetics. So one possible solution to this is this new project, Our Future Health, which I'll just talk about very quickly. As Richard said, initially funded with industrial life sciences strategy funding, co-funded from founding members from industry and charity, some additional funds from NHSX. And the difference here is that this is essentially proposed to be a 10% a sample of adults in the UK. And the aims are very, very translational. So the question is not only how do we discover stuff, but how do we apply it? And we intend to give individual results back to participants. We can talk about how to do that quick answer is we don't quite know yet. And then for results that we can give back, in terms of integrated risk scores, will people be interested in being in risk-stratified studies that might be deep phenotyping studies for a few dozen people with a particularly interesting homozygous knockout variant, or could be tens of thousands of people who are at higher risk of a particular disease in a primary prevention study. These are all of the partners. All of the information is completely public, both industry and charity. The plan, it's a much lighter touch than UKB because of the larger number that we hope to get, but questionnaire, blood, consent to linkage. We'll be doing a genetic array on everybody, not sequencing, genetic array. And we're planning to do cognitive function tests, other activities, all of this linked to NHS records, hopefully including primary care. The array, after a competitive process, an independent board chose Illumina, and of relevance to the pharmacogenetic community, they will include their pharmacogenetic component, their tool, which will cover the 880 variants that Munir and his team had given us for inclusion. We've got a genome-wide backbone, and then we've got custom content that includes a variety of targets. So again, we will make absolutely sure that the variants that Munir indicated on behalf of the pharmacogenetics community, if they're not in the Illumina product, then they will be covered by the array. Lots of PPIE has gone into this, and we have the mandate to enroll a very diverse population, geographically, all 4 nations, and with respect to SES and minority status. We start enrolling next month. We did pilot studies last year, and we'll see how that goes. And with that, I'd just like to thank the guys in my group who did the analyses that I showed you of polygenic risk score applied to pharmacogenetics, and then also thank the team at Our Future Health, a growing team that is doing all the work getting ready to roll out enrollment, hopefully starting next month. Thank you.